it's Mrs. Shannon, and we're going to go through the Chromosomal Abnormalities, prezi by David Nufke. This is part of the information unit that we have going on. You should probably watch this after you've done the initial cell division, so you understand some of the references that are being made here, especially in terms of the synthesis phase or the S phase. So I'm going to go ahead and put this into big screen. And so in this screencast here, in this prezi, we're going to look at how genetic material can be changed. And when we're talking genetic material, we're looking at DNA. And how do the changes in the genetic material of an organism lead to changes in the traits of that organism? And so when we're looking at how these changes occur, and then looking at how these changes are carried on into organisms, we need to look at cell division a little bit more closely. And so we're going to look at cell division focusing on meiosis, because meiosis is when the cells divide in a reductive state, and it passes on these cells to new offspring, this DNA, this genetic material to new offspring in the form of gametes that combine during fertilization. And so one way that the genetic material can be changed from a parent to a daughter cell is through non-disjunction. In non-disjunction, chromosomes do not separate during the anaphase. This leads to gametes with irregular numbers of chromosomes. And so I'm going to go back here. And so we have meiosis. And what happens is, instead of them being pulled away, the sisters here, we have, in this case, both of them going to the left side. Okay. This non-disjunction event happens in the first mitotic cycle. Okay. At that point, we have less genetic material in this one, which leads to daughter cells with N-1 chromosomes. We also have 2 with N+1 chromosomes because it has extra genetic material from this first mitotic stage. In this case, it happens at the second mitotic stage. And remember, in meiosis 1 and meiosis 2, we're going through PMAT. We're going through PMAT one time, and then we're going through PMAT a second time. And so here it's occurring through the second time. And so you have 2 cells here with the correct N number of chromosomes, but now you're left with one from this one that is at N-1 and one here that's N+1. And so keep that in mind. The number of daughter cells with abnormal chromosome numbers is going to differ depending on which stage of meiosis that the non-disjunction occurs in. It must happen in a germline cells to have an effect. And the reason is because these germline cells, these gamete-producing cells, are what's going to be carried on and fertilized during the next phase. Segments of chromosomes can also get altered during DNA replication. Sometimes there's errors when DNA replicates itself during the S phase. When that happens, we can have one of 4 different types of errors. We have deletion. Deletion is a removal of a chromosome segment. So when replication is occurring, an entire section of the chromosome is missing. When we talk about DNA and DNA replication next semester, we'll talk about the actual how replication occurs and some of the preventative actions, these double-checking proteins that are involved to help maintain that DNA is replicated correctly, but mutations and accidents do occur. In duplication, a segment is repeated. So in this case, BC is repeated. So we have deletion, duplication. We also have inversion, where it's flip-flopped, and inversion reverses a segment within a chromosome. So BCD becomes DCB. And translocation removes a segment from one chromosome to a non-homologous chromosome. And what that means is to a different pair. So like from chromosome 2 to chromosome 3. So how do these alterations affect an organism? It really depends on the type of mutation that occurs during replication. So here are some examples. Some examples involve just one single chromosome that has a mutation. And we call these SOMI mutations. It involves individual extra or missing chromosomes, and it's usually results in an early miscarriage, but not always. So in Down syndrome, we have trisomy 21, and due to the non-disjunction, we end up with a gamete that has an extra chromosome 21, and so we have 3 copies instead of the 2 copies that are normally going to be found within a karyotype. They usually result in early miscarriage, but oftentimes they do go undetected. Now, some of you guys may know that a couple of summers ago, we were pregnant with our 4th daughter, and she was actually, she did not make it, and she had Edwards syndrome, which is trisomy 18. So it involved a non-disjunction of chromosome 18 during meiosis, and resulted in her formation and her disorder. Unfortunately, it's one that's not compatible with life. Down syndrome is compatible with life, just quality of life. And so she did not make it to birth. Okay, and here you can actually see 3 copies of Down syndrome. 1, 2, 3 here instead of the normal 2. Results of this are characteristic facial appearances, varying levels of mental retardation, slowdown, and other varying effects. And so what happens is you might have, because you have an extra copy, you have extra proteins that are being made. And depending on which proteins are being made in excess, you will have different symptoms that are appearing, different traits that are going to be expressed in varying degrees. So why would an extra 21 be tolerated by the organism? It's because there are genes here, number one, that are going to create proteins that are not going to be lethal to the organism, and normal life functions can still be carried out. Now, sometimes we have so many mutations in sex chromosomes. They're generally more tolerated by the organism, and they don't generally result in a miscarriage. So you're looking at your X and Y. Turner's syndrome is known as monosomy X, meaning that there is only one X. Phenotypically, they look female. However, they are sterile, and it's associated with heart defects, because also on the X chromosome, you do have genes associated with heart proteins. Klinefelter's is XXY. Phenotypically, they are male because of this Y being displayed. They have reduced fertility, but they have some female secondary characteristics, such as overdeveloped breast, and that's because of this excess X proteins that are being produced. Sometimes you have multiple Y chromosomes, multiple X chromosomes. They're not a problem. Why? Well, in this case, you have XXX. You're just having more of what they already are going to have. And Y here, you have an XSY. It's still going to be male because it's got the Y chromosome. But here's the deal with Y. Is it Y chromosomes? I'm sorry, guys. They are the smallest chromosome, and they have the fewest amount of genes on them. And so the predominant issue with people that are double Ys here are that they may be missing proteins from the X chromosome, but they don't really carry much on them. And so it's not going to be as much of an issue later on. Oops. Okay, and you can actually see here's our X and here's our little Y. Here's X chromosome. Here's an allele for orange. Okay, so the X chromosome does carry genes, extra genes on it. So there are some things that are associated with excess X chromosomes. Calico cats present an example of one of these characteristics here. In this case, you have 2 X's here. You have no Y's. And so each X chromosome is going to display its allele for fur color. If you had an, oops, yeah, I think I just said that backwards, guys. Sorry. Okay, sorry. When you have more than one X here, And I completely skipped this part of it, and I spaced out. I'm sorry, guys. When you have more than one X, so in us or in other organisms, generally one of the X's is inactivated. It's turned off early in development, and there are proteins that do that. And the reason is so you don't have overexpression of certain proteins. Sometimes, however, the X gene is not completely inactivated. And that's what happens with calico cats. So here we have early in development. Normally, one of these would be turned off and become what's called a bar body, which means inactive. However, in calico cats, the one that becomes inactive is going to differ depending on which cell in the body. And so there are going to be some cells in which the X right here with the orange phenotype is inactive and becomes the bar body. And so the black fur is going to be displayed. There are other cells in which the black fur is going to become the bar body, the inactive form, and the orange is going to become the one that's displayed. And so you're going to have orange fur displayed. And this is how we get calico cats. Calico cats are always female, and it's because of this inactivation of one of the X chromosomes. Now, sometimes we have polyploidy mutations. These are whole sets of chromosomes. They involve extra homologous sets. They're not usually tolerated in animals. If you have a whole set, like double everything, triple everything, very early on, these lead to miscarriages. There are sometimes rearrangements. There's no rule about how a chromosomal rearrangement will affect an organism. There's a lot of let's wait and see. Let's see how this happens, because it depends on the chromosome. It depends on which genes are rearranged. It depends on if it happens in all of the somatic cells or just a few of the somatic cells. Depends on if there's other genes there to compensate, et cetera, et cetera. So a translocation, just so you know, though, in blood stem cells leads to what we call CML leukemia. And so some of them are known, but some of them just aren't known. So here's a map showing diseases associated with various translocations in the human genome. And so here we have one from 5 over to 2, anaplastic large cell lymphoma. Schizophrenia is when we have one from 11 that's translocated onto chromosome 1. Let's see, what else do we have? There is a form of Down syndrome that we go from 21 over to 14. There is infertility associated with translocations between 13 and 14. And so these are just a few of them. A lot of them are unknown and wait and see scenarios. So at this point, you should be able to make sure that you can explain how the mutations that are discussed in the presentation arise. The big ones being non-disjunction which can occur during either mitotic phase of meiosis or errors during DNA replication. And there were 4 different types that were discussed. You should also explain the causes and effects of all the situations discussed in this presentation. So you should be able to explain what happens when you have one extra chromosome, like trisomy 21 or trisomy 18. You should be able to explain the importance or significance if you have extra X or Y chromosomes. And you should be able to explain how when you have more than one X chromosome, one of those are going to be inactivated and become the bar body to prevent overexpression of a gene on the X chromosome. If you have any questions, please let me know.